Yeah, good morning, everyone. Thank you very much, Ingmin, and thank you very much for the invitation. And really thank you to the audience for being here at 8 a.m. today. I'll try my best to make it worthwhile. So I'd like to talk about our recent work on non-invasive optical methods for the assessment of cerebral hemodynamics using diffuse optics and the new technique that we have recently developed that we call coherent hemodynamics spectroscopy. So I will first by introducing the basic ideas of both coherent hemodynamic spectroscopy, or CHS, and the quantitative hemodynamic model that is strictly connected and associated with it, and how this can be used to interpret the phase difference between the oscillations of oxygen deoxyhemoglobin concentrations that are measured routinely with near-infrared spectroscopy. And then I will show how this technique can be used to measure absolute cerebral blood flow, CBF, and how, by taking advantage of the continuous measurements of NIRS and the quantitative hemodynamic model, how we can also continuously monitor cerebral blood flow using this technique. And of course, this is relevant on its own merit, but it is also a tool that will allow for cerebral autoregulation measurements. Autoregulation is the process that maintains a relatively constant CBF even in the presence of changes in perfusion pressure. So here are the basic ideas of coherent hemodynamic spectroscopy. The idea is to perform measurements of coherent dynamics of oxy and deoxyhemoglobin concentrations, which we measure routinely with NIRS. This can be oscillations as a function of frequency, that's the frequency domain, CHS, or these can be measurements of transients as a function of time, and this is time domain, CHS. So oscillations are present or can be induced. Transients can be associated with challenges or with functional activation. So even on its own, these measurements will provide information about the hemodynamics in the brain, and this information could be correlated with physiological or pathological states. So this would be a model-independent approach, but one additional step, one new dimension is introduced by an analytical model that relates oxygen deoxyhemoglobin concentrations to physiological quantities such as blood flow, blood volume, and metabolic rate of oxygen. So the idea is to use this model to translate CHS measurements into meaningful physiological parameters. So all of this is described in detail in these two publications in the special issue on FNIRS, on neuroimage, and in physiological measurement. So those are the basic ideas of coherent hemodynamic spectroscopy. The analytical model, I'm not going to describe it in any detail. Again, this is the publication where every step of the model is described in detail, but there are essentially two ideas that are behind this model. The first one is to follow the flow of red blood cells and all of those hemoglobin molecules that are inside the red blood cells in the smaller arterioles and capillaries where oxygen diffusion to the tissue happen. And just quantify the desaturation of hemoglobin in this flow by using two parameters: the capillary transit time, the mean capillary transit time of blood, and the rate constant of oxygen diffusion from blood to tissue. So the model obtains an analytical and quantitative description of the desaturation of hemoglobin in response to a step change in blood flow velocity. Now, of course, this is a very special case. It's a case of a step change in flow velocity. The second idea is to consider microvasculature as a linear time invariant system, something that in some cases is reasonable and is justifiable, in other cases may not be, but this is the step that is required to translate the step change response into an arbitrary response to oscillations at any frequency to the transfer function, or to transients associated with any perturbation through the impulse response function. So these are really the two key ideas. Quantify the desaturation of blood in the capillary compartment and treat the microvasculature as a linear time invariant system. And just to give you a sense of what happens, if this is the step change in flow velocity in the capillary compartment, this is the time response of the hemoglobin saturation in the capillary compartment. So to go from the first to the second steady state, there is a finite time required, and that is true also on the venous side. Even though the detail of these transits are different, still there is a time constant associated with the microvasculature to respond to a step change in flow velocity. And of course, as expected, this time constant is directly related to the blood flow, to the blood transit time in the capillaries and in the veins. So this is really what the model does. It calculates quantitatively this hemodynamic response and then obtains transfer functions and impulse response functions that allows for the generalization to any arbitrary perturbation. So this means that now we can really interpret the phase relationship between oscillations in oxygen-deoxyhemoglobin. This is something that has been reported in the literature for 20 or 30 years. And over the past 5 or 6 years in my group, we have really hit our head to figure out the underlying origin of these phase differences. So in the case in which you have a blood volume oscillation, let's say something like this, then oxygen-deoxyhemoglobin will oscillate in phase with the blood volume oscillations, in phase with each other, with an amplitude relationship that depends on the saturation of the volume oscillating compartment. In the case of blood flow oscillations, or I should point out, this could be also metabolic rate of oxygen oscillations, CBF and CMRO2 are intrinsically linked in their effects to NIRS studies. For the remainder of this talk, I will assume that CMRO2 oscillations are negligible with respect to CBF oscillations, something I will comment on again later on. But if you have CBF, blood flow oscillations, now oxygen-deoxyhemoglobin oscillations are going to be exactly out of phase, in opposition of phase with each other, 180-degree phase difference. They will have the same amplitude, but, and this is the key thing, they will be phase shifted, phase delayed with respect to the driving blood flow oscillations. So this means that even if blood volume and blood flow are exactly in phase with each other, as assumed here, then oxygen-deoxyhemoglobin may have an arbitrary phase difference as observed in the literature. So I really think this is probably the most important slide of this talk because it provides a key to interpret the phase difference between oxygen-deoxyhemoglobin oscillations or time delays between them. So this may be the most important slide, but it's not the most exciting. The most exciting is coming up, and I'll point out when we get there. But this is a case where blood volume and blood flow are assumed to be perfectly in phase with each other. It turns out that this is almost never the case. And in fact, because of cerebral autoregulation, blood flow usually is ahead of blood volume changes. And this can be represented by a relationship between blood flow and blood volume. And here I should point out that autoregulation is really a relationship between blood flow and blood pressure, mean arterial pressure. But in a number of cases, it's reasonable to take blood volume as a surrogate for blood pressure. So in a phasor representation, this is much more conveniently shown. These are vectors whose amplitude and phase represent the amplitude and phase of sinusoidal oscillations at angular frequency omega. So CBF, blood flow oscillations lead oscillation in blood volume. So this is really the framework for all of the studies that we're doing based on EFR spectroscopy and coherent hemodynamic spectroscopy. So let me show now some results. The first study we did was on healthy subjects. I mentioned that here we need to work with coherent hemodynamics. So hemodynamic oscillations that have a well-defined amplitude and phase relationship between them. So one approach we're following is to induce systemic blood pressure oscillations, and they're going to induce hemodynamic oscillations everywhere in the body, but of course the brain is where we're going to measure them. So this is where we do our measurements in the prefrontal cortex, and in this case we induce systemic blood pressure oscillation by pace breathing. So we ask the subjects to inhale and exhale at a paced frequency given by a metronome. We use 10 different frequencies to perform coherent hemodynamic spectroscopy, frequency-resolved studies of these oscillations with periods from 5 to 14 seconds. And everything is reported in these publications, again, in the special issue on functional NIRS, in neuroimage, and in GEOS. So once we perform these measurements of amplitude and phase of hemodynamic oscillations at these induced frequencies, we obtain spectra, which we call CHS spectra, of phase. These are phase difference between oxy and total, or between deoxy and oxy, or amplitude spectra, amplitude ratios between oxy and total, or between deoxy and oxy. And as I was saying before, these spectra are model independent. Those are just observables that can be related to physiological or pathological conditions. But now the additional step, the next level, is to perform a fit with the hemodynamic model. And so now we can get quantitative measurements of the fitting parameters, which are reported here. And they are the capillary and venous transit times in seconds, the cutoff frequency for autoregulation, a measure of the effectiveness of autoregulation, and other parameters that are related to the blood volume in the arterial, capillary, and venous compartments. But let me focus now on TC, the mean capillary transit time. This now can be translated into an absolute measure of cerebral blood flow. And the way it is done is by using the central volume principle, where cerebral blood flow is given essentially by the capillary blood volume divided by the capillary transit time. 1 over rho b is the rho is the density of brain tissue, is just there to convert CBF into the standard units of milliliters of blood per 100 grams of tissue per minute. Rho is about 1. It's typically 1.04, 1.05 grams per milliliter. But the point is that this capillary blood volume can be expressed in terms of measurable quantities. The total hemoglobin concentration in tissue, which is measured, for example, with frequency domain or time domain near-infrared spectroscopy. The concentration of hemoglobin in blood, which may be assumed in a healthy population or may be measured by drawing blood in a clinical population. And then these are the two key parameters, the capillary blood volume fraction and the capillary and the blood transit time in capillaries. These are the two parameters that are measured quantitatively and non-invasively with CHS. And with this, in the case I reported, we obtain a cerebral blood flow of about 90 milliliters of blood per 100 grams of tissue per minute, which is comparable to the normal value in gray matter, which is about 80 in those units. So this is a method to measure absolute values of cerebral blood flow, and we have applied this in a clinical environment. In this case, to induce these systemic blood pressure oscillations, we did not use paced breathing, which requires significant subject cooperation, but we used a method that is widely used in the literature, which is a cyclic inflation of a pneumatic thigh cuff, which is inflated and deflated at above systolic pressure. And in this way, we perform measurements on 6 healthy subjects and 5 patients undergoing hemodialysis treatment. And what we found is that the absolute value of cerebral blood flow was significantly lower in the dialysis patients with respect to the controls. And again, these measurements are done in absolute units of milliliters per 100 grams per minute. So these are absolute measurements of cerebral blood flow. This is something like a baseline, a snapshot measurement. But we can take advantage of near-infrared spectroscopy, the continuous monitoring that can be done, and also the relationship, the analytical relationship provided by the model between NIRS signals and cerebral blood flow, blood volume, and oxygen consumption. So we decided to do this in a protocol which is also widely used in the literature, also involved pneumatic thigh cuffs. In this case, they were kept inflated for 2 minutes above systolic pressure, in this case 200 millimeters of mercury, and then they were quickly released. The effect, again, is a systemic perturbation to the mean arterial pressure, which drops. This is the drop of about 10 to 20 millimeters of mercury. And then correspondingly, we see the coherent response of the oxyhemoglobin decrease and deoxyhemoglobin increase measured in the prefrontal cortex with NIRS. So this is the time period that we're looking at. That is the transient associated with the perturbation in the mean arterial pressure. So let's focus on this. And this is what was done in 1989. And since then, there has been a large number of papers in the field of transcranial Doppler ultrasound mostly to measure autoregulation. So this is the transient drop in mean arterial pressure, in this case by about 20%. And this is cerebral blood flow measured with transcranial Doppler ultrasound. So these measures are done through a temporal window by measuring the flow velocity in the middle cerebral artery. So these are measurements done on a large blood vessel. And in this case, what we do instead is, again, the same kind of transient drop in arterial blood pressure. This is measured now with near-infrared spectroscopy and coherent hemodynamic spectroscopy. And now this is the cerebral blood flow trace measured non-invasively, continuously, and in absolute terms with near-infrared spectroscopy and CHS. So here again, now we have absolute numbers of the y-axis and continuous monitoring. And so we can now monitor this decrease. So to me, this is the most exciting slide of the talk. I think this is the first time that there is a measurement that really achieves all desirable features of non-invasive measurements. There is no bolus here. There is no injection, no blood sampling. There is no calibration measurement done to obtain these absolute values. And it performs continuous monitoring of cerebral blood flow. These measurements are important because, as I was mentioning before, they can be used for autoregulation assessment. In that case, the critical window is this window of about 5 seconds. That is the window where blood pressure remains about constant at the lower value, as induced by the cuff release, before baroreflex phenomena starts correcting for this. So this is the useful time window where you can see the increase in blood flow, cerebral blood flow, obtained by autoregulation. And this rate of increase provides a measure of the effectiveness of cerebral autoregulation. So the same thing can be done here. That's the 5-second useful window. And this is where we have an approximately constant mean arterial pressure before the recovery through baroreflex effects, which really are confounding effects to autoregulation. But this is the kind of rate of change that we are after. I should also point out that a couple of things. Diffuse correlation spectroscopy also can perform this kind of relative measurements. They're not in absolute terms, but they are non-invasive and they provide this kind of real-time monitoring. I should also point out that the two techniques are really conceptually different. DCS is based on the decorrelation associated with the fluctuation in the near signals because of blood flow. Here instead, this technique is based on the central volume principle. I should also point out that DCS really, truly measure cerebral blood flow. In this case, I had to make the assumption that metabolic rate of oxygen changes can be negligible. When this is not the case, for example, in functional activation studies, the two techniques, DCS and CHS, could be usefully combined in obtaining a true quantification of both CBF and CMRO2. So, but continuing on this line of thought, on this kind of perturbation and the useful 5-second window here to study cerebral autoregulation, we have performed a study on healthy subjects. Again, the blood pressure perturbations were induced by cyclic inflation of thigh cuffs. And here there is the 2-minute inflation of the cuff pressure, fast release, the resulting transient decrease in mean arterial pressure, and the measured oxy and deoxyhemoglobin concentration releases. So there we did measurements during normal breathing, normocapnia, and during hyperventilation, which induces hypocapnia. And it is well known that hypocapnia increases the effectiveness of autoregulation. So these are the results we obtained. These are the average results on all of the subjects during normocapnia. These are the top and bottom traces, and during hypocapnia, which are the middle traces. And here you see that, again, the useful window are the 5 seconds between time 5 and 10 seconds here. That's when you have the differentiation of autoregulation, and you have this faster recovery during hypocapnia. The symbols are the experimental data obtained with NIRS. The lines that really go to the point perfectly are the fit to the hemodynamic model. And the fit to the hemodynamic model, in this case, provides an absolute measurement of autoregulation through the cutoff frequency. So these are the results on the 11 subjects measured. And during normal breathing, the dark bars, we always measured a lower cutoff frequency for autoregulation than during hyperventilation-induced hypocapnia. There's only one exception, subject 9, who we measured 5 more times to be sure, and every single time this subject showed a decrease. So of course we don't know why, but we completely trust this result. We also measured 4 more times a number of subjects, and we always reproduced the findings. So there really may be something here, and those are some of the things we are exploring at this time. So really that's what I wanted to say. I have shown a new hemodynamic model that provides analytical relationships between the oxygen deoxyhemoglobin concentration measured with NIRS and the physiological quantities, cerebral blood volume, blood flow, and metabolic rate of oxygen. I presented coherent hemodynamic spectroscopy as a method to perform time-resolved measurements, either in the frequency or time domain, of coherent oscillations of oxygen deoxyhemoglobin. And then I've shown how coherent hemodynamic spectroscopy provides non-invasive, local measurements of cerebral blood flow and cerebral autoregulations. One of the main efforts currently are to perform depth-resolved CHS to achieve discrimination between the top layer of extracerebral tissue and the bottom layer of cerebral tissue. Of course, I really need to thank my group, in particular Bertana Hlacholu, Michele Piero, who have been the first PhD students really translating these ideas into human subjects. Christian Javalekos, she's the PhD student working on that now. Angelo Sastrolis, my partner in crime for everything that is going on in the lab. And Jana Kainerstorfer, who has completed her postdoctoral period in my group and is now a successful assistant professor at Carnegie Mellon University.